So, good morning, everybody. It's a pleasure and honor for me to be here in front of you and to go through this third edition after the first two in Italy. Finally, we moved forward after the pandemics. And my talk is about applied biotremology, which is a bit the common topic of the morning. And to show how it's possible to join very basic research to applications that are useful, especially for farmers, but not only, for many other aspects that are important in everyday life. And let's see if it works, this guy. I don't know.
Can you share it again?
Okay.
Sorry.
Just do this, because I have no idea. I was supposed to do this.
This one or this one? This one.
Share sound.
Share sound.
I hope it's the right presentation.
This one is not plugged in. This is the plug-in. Sorry. Like that. And now to turn it on.
Okay.
Yes.
Okay. So I want to tell that applied biotremology and biotremology in general is not something really new because, as you can see, it was in the Encyclopedia, 1773, and there is a part about aneton. Aneton are the May beetles, and here it's written in French, but the translation is that by shaking the trees with the arms or with the poles, you can collect May beetles. And you can put them in a basket and then boil them in the water. That was the suggestion. So basically, shaking a tree is biotremology, okay, applied biotremology. And things went forward. Here we are in 1889. This is an Italian manual from southern Italy, from Naples. And basically, the concepts that are expressed here are that we are talking about, I think, apple orchards, but the point is that if the trees are young, a single blow with the palm of the hand on the stem is enough to make the beetles fall. So you can imagine like karate sing. But if the trees are large, to vibrate the foliage, it is necessary to use an iron, lead, or wooden hammer covered with rags of clover because you don't want to damage the bark, of course. So you can imagine these people hammering the trees. But if the trees are very large, that's interesting. You need to send some boys up. But the very interesting point is that you should be barefoot because you don't want to ruin the bark again. So it's very important, different techniques according to the size of the tree. And this is still applied biotremology, indeed. So now we'll go to something a bit more serious. And new technologies. This Journal d'Agricultura Tropicale. This is a French review. Here probably I'm covering the title, but it's about fourmis blanches that are the termites. And this is a curious application, a curious application, the telephone. This is the telephone in that time, and you can see it's a very old-fashioned stuff. And here the point, I put some, this is French, my French is terrible, but par un véritable téléphone, you have microphone, tube d'acier, and qui révèle presents the fourmis blanche. So this is a termite detector, okay? And the point this was called Termitenzucker. This guy is Carl Leopold Escherich. This is the inventor of this Termitenzucker. And Escherich, by the way, is like homonym of the guy of the E. coli, the Escherich. They're not brother neither, but however, they were living in the same period. And this guy, you see in the picture, they were working in the colonies in Ceylon, German colony in that time, and termites were terrible. And the point was to look for them in the ground, in the trees. And in the picture, actually, they're using another device that is the termite killer. Basically, they're pumping down in a nest after detecting it with the Termitenzucker. I don't tell what's inside that poison, but now would be super forbidden. However, the description is it consists of a microphone inserted in a funnel at the end of a steel tube connected with a telephone receiver. So you can imagine these people with the telephone going around. And then human ingenuity and modern technology are well on the way to eliminating the fear of the white ants. Basically, after 100 years, we're still in the trouble with them. But it's nice, this modern technology. But every time is a modern technology when you do something new. And this is basically how it works. You see there is the structure typical. You have the funnel is the amplifier. Then you have the sensor. That is the loudspeaker, basically. You have the loudspeaker on the opposite side. That is the receiver of the telephone. Then the tube still is the acquisition device. Then you have the infested wood. But this is a picture 2022 is exactly the same thing. So actually the principle that were used in 1911 are still today valid. And you say, oh, we didn't improve much. Well, actually, we improved, technology changed, but apparently we didn't solve the problem of the termites. So now I'll go in something a bit newer. Unfortunately, the title is covered. I don't know. It's 1955, 54. Oh, this is a thesis of Pesce from Colorado. And this is a bit... started animation, okay. Well, however, the point is now we are doing something a bit more complex because this is a unit of 4 boxes. And one with the other, you see there are different compositions, sizes, materials of these boxes to prevent different kind of interference from the background noise, from electricity, from sound insulation. And you see, this is the pictures, and the point was to detect the presence of weevils inside the grain, the seeds. And I read the thesis, it's very nice, and it's even rather modern for the time. After that, in the 60s, there was an explosion of inventions, of new solutions. So on this one on the left, a mechanical method of retaining flying insects on a test surface. Maybe it could be useful even today. You don't let them fly away. You just put a vibration so they can't jump away. They stay there all the time. And the other one, 66, artificial acceleration of hatching in quail embryos. So you see that this idea that it's possible to condition hatching using vibrations was already on the air in 66, and nowadays we are working on it about synchronization of eggs, acceleration of eggs hatching, and you see also the paper of Karen Varkentin. So you see that this was already a topic, in this case birds, but interesting invention is also this incredible machine with, I don't remember if 18 or 16 spaces where you put single seeds, and then there is the needle of a play recorder on each of them, and then there is an oscillogram, and then you can see where are exactly the infested the infestation, in which single seed you have a weevil. So these are in the 60s. But now we come to something that is, for me, was inspirational, and I got some ideas from this. That is the first attempt of mating disruption. 1979, Saxena and Kumar from India. The title is Interruption of Acoustic Communication and Mating in a Leafhopper and Planthopper by aerial sound vibrations picked up by plants. So basically transformation of airborne sounds in vibrations. And as you can see, here there are 2 targets. That is a leafhopper and a planthopper. They are very important for economy, Marasca devastans and Nila parvata lugens. And as you can see, they try different types of instruments, music instruments. This is the harmonium you see on the right side. Harmonium, sort of piano, or, I don't know exactly what, but it seems very old-fashioned. And they saw a couple of important things indeed, that the frequency is really crucial to success because, I don't know if I can, yeah, okay. No, I can't move from here, but you see that these are the percentage of mating, and you see that 10 is in correspondence of 200 Hz or 300 Hz. While if you change even slightly the frequencies, you have a reduction, a progressive reduction of success in mating disruption. Also, the amplitude is very important. So you need the threshold of amplitude, otherwise it doesn't work because you need to mask this signal. While other instruments apparently didn't work so well, but the trend was confirmed also for other instruments. So probably this harmonium had some special features that made it easier, the transfer of airborne sound to the plant as vibrations. So here we go to the concept of behavioral manipulation. And nowadays, it's common in many papers, and many industries are searching for solutions of behavioral manipulation. Definition is achievement of pest management through deliberate manipulation of a pest's behavior. We have seen some example, we will see some example today. And manipulation, of course, the use of stimuli that either stimulate or inhibit a behavior and thereby change its expression. That's the definition given by Foster and Harris in 1997, which is an important paper as a reference for behavioral manipulation. And what are the most common elements of behavioral manipulation? Well, semiochemicals. Semiochemicals are largely used in agriculture nowadays, many different fields, rice, wheat, apple orchards, peach. You find almost everywhere semiochemicals as pheromones in particular. And these are chemical substances or mixtures that affect the behavior of other individuals. An example, grapevine moth, Lobesia botrana, which is a bad pest in southern Europe, also in California for some years, but then it was eradicated as much as I know, thanks also to behavioral manipulation approach, but also for bad pesticide usage. However, consider how many applications are possible, because you can use pheromones in different ways, different rates of release, different in density, different aims, like you can do monitoring, false trail, mating disruption. So you see that semiochemicals are employable in many different ways. And this is an example of an apple orchard. You see that there are 9 target species with the pheromones only in the apple orchard, and tons of different traps that are specific for each species. So you understand that the potential of using behavioral manipulation, it's huge. And this is just one crop and one order. Of course, Lepidoptera we know are maybe probably the best user of pheromones, but they're not only. Okay, and recently we introduced the term of semiophysicals. We can define them as physical signals, both mechanical and visual, that affect the behavior of other individuals. So we just changed the first part of the sentence, replacing chemicals with physical. And actually, there are 4 types of semiophysicals, because lights, colors, sounds, vibrations. Of course, here we talk about vibrations. And I want to go through a parallelism between semiophysicals and semiochemicals to show how they can be interchangeable and how definitions that are common nowadays about semiochemicals can be moved, shifted to semiophysicals without changing anything, basically. So you see that we can we can split between intraspecific signals, interspecific signals. And intraspecific signals, you have mating, parental care, rivalry, territoriality, alarm, synchronization, recruitment, aggregation, and others. They're not the only one. Interspecifics, we can divide in 3 categories: benefit for the receiver, detriment of the receiver, benefit of both sender and the receiver. These are defined nowadays. The first category of intraspecific signals are pheromones in chemical ecology, while the other 3 are chiromones, allomones, cyanomones. These are common terms for people that work in semiochemicals, okay? But I can show you that in semiophysicals, we can do exactly the same thing. So intraspecific signals, mating behavior, okay. It's the most common thing that we have vibrational signals that mediate the mating behavior. Parental care. There are papers, just an example here, about the wasps. That there is the double interaction between the larvae and adults. Adults can emit signals, and then the larvae by themselves respond with other signals. So this is a parental care example, but it's not the only one. And egg hatching synchronization. That's another example of intraspecific communication at the embryos level. But also, we have example of benefit for the receiver. Predators, they eavesdrop the communication that could be incidental, accidental, or volunteer when they are more species-specific. Generalists, like intercept, probably just walking, grooming, and other activities that are not specific. But also parasitoids eavesdrop. And we see with Lorenzo a few days ago, or yesterday, I don't remember exactly, that they can even interact with the larvae inside the berry. So these are classic examples where the eavesdropper has a benefit from receiving a signal. But we have also benefit of the sender. This is an example of a paper of Casacci, Barbero. It's about this lysannid butterfly. The larvae live in the ant nests. But normally, ants will kill them if there is not protection. So they emit vibrations that are mimic of the queen. And in this way, they are not only, I would say, ignored, but even protected and fed by the ants. So ants take care of a parasite. So you understand that in this case, the benefit is to the sender. Another example of benefit of both is buzz pollination, because in this case, as you know, by the way, I didn't see paper speech about this, but this is an interesting topic. However, in this case, by emitting a specific vibration by a bumblebee or another kind of bee, you have a benefit for both the sender, which is the bee, and the receiver, in this case, is the flower. For the pollen, it's released at higher numbers. And this is an example from Hager and Krause about the multi-actor communication that was presented in Riva del Garda. And it's about ants, acacia trees, and elephants. Probably most of you read the paper, a very interesting paper. And as if you remember, I just try to explain the figures, but wind and other abiotic factors induce vibrations that are aspecific and are not really considered by ants. We are in the savanna here. And however, when a big herbivore, like an elephant, is eating from the tree, you see from the vibrations that are higher in frequency from the figure, this triggered the reaction of the ants, and the ants are aggressive and start biting the big elephant. One can say, oh, okay, but just ants and elephants. Well, elephants can be scared by this, and so Hager and Krause also demonstrated that the presence of nests on the acacia trees favor the survival and diffusion of the tree. So this is a multi-actor where there are benefits for some actors and detriment for others. But these are just examples to show that whatever I showed in the previous picture with the definition of semiochemicals, pheromones, chiromones, whatever, is perfectly applicable also to biotremology. And in fact, I'm here to propose new terminology. And after semiochemic physicals, now it's time probably to use another term from the Greek that is doni. Doni means to vibrate. So a possible, I didn't write it anywhere for the moment, but a possible option is to use for intraspecific signals pherodones or intradones. That is, sounds good. No, pheromone, pherodone. But you can have also this other solution, chirodones, well, the benefit of the receiver, allodones, to the detriment of the receiver and benefit of the emitter, and synodones. I guess that I should present this in a conference of semiochemical people and see the reaction, because here maybe everybody agrees, oh yeah, it's a good idea, but I need to talk to them. But however, we don't need their permission to do this. The point is that I don't see a reason why we shouldn't do that, because I just show you that it's perfectly applicable according to their definition. So I don't see a reason we shouldn't do that. So going back to this slide about semiophysicals, we have 3 different approaches that are possible. Attraction, repellence, I put slash because attraction and repellence are the opposite to each other, but promotion and inhibition, and disruption. Disruption is a bit borderline because it could be a sort of inhibition, but let's see. You see from this slide that basically on the left we have target species that are, I just put the most common ones and from the paper that I'm reading, but there are others, of course. And also I put the vibroscape because this is a nice application that could be transferred to ecology, but also to agriculture when you monitor insect pests. If you are specifically targeting some species, it's easier also for the artificial intelligence and deep learning if you have to concentrate on 1, 2, or 3 species. But on the other side, you see that you can also target many different behaviors. And in the next presentation this morning, you will see cases that are reported in this slide. And then you can see applications and ideas that are included in this scheme. And there are many others that are not here. There are many others that are to come. Because many groups, like the one in Japan where Takuma works, well, you have a presentation about this, so you will see. So this is basically a scheme that people working in applied biotremology should follow. I simplified it, but basically you have 3 steps. First of all, identify the target behavior, because even in leafhoppers, it's not possible to have the same approach if you're working with one species or another one, because the mating behavior could be totally different, and the role of the signals can change a lot. So you need to find what is the target behavior and associated signals that are targetable. Then you have to work on the signals. Yesterday we saw presentations about how we can handle signals in playback tests, also in testing the efficacy of them and see what are the different options, and then use them in trials and bioassays in the lab, in semi-field conditions, and also in open field, because until you don't go to the open field, everything is rather theoretical, but well, if it works in the lab, it's a big question. Does it work also in the field? And we have the enemy that is the background noise, human interference. So it's not something easy to transfer, but it's possible, I can tell you. So I want to talk about our main target species model of our research, that is a leafhopper, the grapevine, American leafhopper, and is an invasive species from the 60s in Europe, the Scaphoideus titanus. It is a problem because it's a vector of a phytoplasma, flavescens doré, and Imane Akaso will talk about this in the next presentation more in detail. But the point is that plants that are affected by flavescens doré, that is the disease, must be uprooted, and this is considered for many years a quarantine species. So it's a big trouble for farmers, and still in these days is a trouble, because in the last years the situation is becoming worse and worse. There are many reasons about that, but let's talk. So what we did, we started in 2006, I would say maybe 5, with working with the Meta in Ljubljana, and I would say that nowadays we recreated the collaboration with the industrial partner, a commercial prototype that is ready for the market, more or less. Then we can ask. And so it took about 15 years, 16 years. And we started from a technology readiness level that was 1. That means really we didn't know anything. And you see that in 15 years, it is really a reasonable time indeed for research, maybe less for industries, but for research is something affordable. But after this experience, probably we are faster now. But this was the first attempt. And you see that we went up to the stairs of the technology readiness level. So I'm going to explain what we did in the next slides to show you how it works. It worked. Okay, early steps. You see that we started with the basic research in the lab. You see that the lab, the first lab was really a room with everything was very arranged in a, let's say, in a very basic way. And the first papers where we found that it's possible to disrupt in condition of lab the mating behavior. Then we moved to the semi-field, and we tested in cages, in sleeves, so in protected environments, and we found that still it worked. And finally, Jernej did this part of the project until 2016. We moved to the real field, still using sleeves, but in conditions that were not different from the commercial situation in a vineyard. So this is a video. Let me see it works. Just to show you how it works, the courtship part of this mating behavior. So this is the buzz. Then you have pulses. And double pulse. There is the female reply. And double pulses of the male. It's like a chicken, you know? And so the courtship normally is about 10 minutes. We are almost in the end. So, I can't even stop, there is the mating, but unless you're eager to see the mating. Yeah, here we go. So, okay. Oh, enough. So basically the scheme is more complex. What you saw is just this. But we described the full mating behavior. And we can say that there are 4 main steps. The first one is call and fly, that's typical of any leafhopper. Then there is identification part, a location part, and a courtship part. And each of them is mediated by a different type of duet and different types of signals. So it's a pretty complex behavior. And there are important rules about the temporal pattern. If they're not respected, the mating is interrupted. So there is a time window in which the female is supposed to respond. That is about 30 milliseconds. Otherwise, the mate stops. And any mistake, as I say here, causes the stop. So it's a strict, I would say, a very strict rule duetting behavior. And we have a hard time to simulate it in the lab because in the milliseconds, the males take decisions. And this is an example. You see that the first part, they are on different leaves. There is a male call. Then there is identification duet where they have this temporal pattern scheme. And then when the male say, okay, this is the female, start moving and searching here. And the location duet is similar, but with a different temporal pattern. And then you have the courtship part when they are only on the same leaf. So the activation of the highest frequency, because the buzz is about 1,000 hertz with harmonics. And so we know from also the previous days that using high frequencies in the distance doesn't make sense because you have attenuation and the loss of information and the energy waste. So basically, they are aware to be on the same leaf of the female at some point. And this awareness is probably due to the amplitude, to the reception of the signals and the comprehension of the amplitude. And this is, I will go back to this concept in the next slides, but just remember that. So this is an important element that we found, is the disturbance noise that is emitted by rival males to stop the ongoing duet of another pair. And this signal, as you can see from the spectrogram, is basically aiming at masking the male signal. So in this way, they really cover and mask the signal of the other. And this causes immediate interruption of a duet in nature. So basically, you can imagine the vibrational mating disruption approach. In nature, you have a situation of a male and a female, and the male calls and the female replies. In case of presence of a rival male, this emits this red sound, vibration better, that is the disturbance noise. And this causes immediate interruption of the communication. So what we did was simply to remove the male and put an emitter of disturbance noise continuously in the vineyard. And this caused the suppression of mating. Well, the concept was this. And you see from here there is a sort of small animation made by our engineers. But the point is to vibrate a pole in the vineyard, and the interference is transmitted along the rows to the plants. You can see from the red part here are parts of the plants that are covered by the disturbance noise. So, however, before getting to that, we also studied many other parts of the mating behavior. One of these was the importance of the signal for directionality. So you can imagine that for a male here, it's a challenge to find a female in a situation where you have many forks, many decisions, different, and basically, if you go random, you will never find your mate. So you need directionality. We tested this in this paper of polliner, and you see that actually we found that there is a directionality. Correct decisions are significantly higher. We made tests in the lab, of course, to see this, and we found that they can have a directionality. That was important also because if it was a random situation, it's hard to make a disruption. So then we studied also the signal attenuation, and we found the thresholds of efficacy of the system. And for instance, this is a paper, this is basically the review paper in the book of 2016, where we presented the active space of Scaphoideus, and importantly, that between the leaf where the sender is, that is this one with the red and the green, immediately after the signal gets out from the leaf where the emitter is, you have a drop of about 10 decibel, and then always. So basically, we found that there are gradients along the plant, but the only place where you have a certain amplitude is the same leaf where the emitter is. All the others are at least 10 decibel under. And we made this scheme where decisions of the male are mediated by the amplitude, perceived amplitude of the female signal. So basically, Karshit duet is activated every time this value of this is a scale in 10 minus 5. It's about, I would say, 10 micrometer per second of velocity. And if this value is passed, they activate the Karshit duet. If it is under this level, it's call and fly. If it is in this way, in this range, location duet. So you understand that it's very important, the amplitude perceived. Another important study that we did was also understanding that it's possible interplant communication. And interplant communication was not recognized previously because the basic dogma was that continuity of the substrate is crucial to achieve efficacy in communication. But we demonstrated that up to 6 centimeters of distance, this is the scheme, you have 50% of overlapping surfaces, like you can see, and the male and female, although with different decisions according to the intensity. Okay, you see when they are very, very close, it's even possible to start the location duet. Karshit, no, because it would need contact Karshit. You have to be on the same leaf. But then you see that the 5 centimeters, they still do call and fly. They activate the call and fly, okay? And some also even at 6. But you understand that 6 centimeters, it's a huge amount. It's like, so this means that active space in the plants, like a vineyard, in the vineyard, can be important. This is another paper that in the field and semi-field conditions. And here we tested the importance of the distance from the shakers and how the signal decay, attenuate with the distance, and how the efficacy in making disruption changes with the distance. So basically, in this article, you can see that within, these are 940 centimeters, so within 10 meters from the emitter in field condition with the real plants. And we had basically very good results, almost 100% of suppression of the mating. And as you can see from the graph, because in any case, even at 10 meters, that is this one, we were in the range of the thresholds. So this one, VL, is the threshold on the same leaf. So when they activate the Karshit duet, at the higher possible energy they can receive in the vineyard. And if we are over these values, then you achieve a mating disruption. So the challenge is to extend to more than 10 meters. And this was our challenge in the beginning, because we couldn't go much further. Because whenever you go, I didn't take the graphs, but if you go to 20, 30 meters, you go down. I don't know if there is a presentation, maybe Iman is doing this. You will see that you drop the efficacy. So amplitude, it's a very important, frequency. You see that 200 hertz, that is more or less, as we have seen previously, the dominant frequency of the disturbance noise. Well tuned with the vineyard, with the grapevines. So we can transmit it for long distances. The problem is the amplitude. Okay. So, and this is a work that Jernej did also to understand which time of the day we should use. Because if you use solar panels, then you work basically in the day. But what's wrong with this? Well, in these experiments, you see on and off of the system. So in black, it was always on, like in this positive control. And you see that virgin maize that we were collecting from the field were almost 100%. Then you say, oh, what about the other 5% or 3%? Well, if you work in a single vineyard and all around there are many other vineyards, it's possible to have movements, migrations. The control, the negative control, you see that almost all animals were mated. So we tried different types of on and off solutions, but basically we can probably turn it off in the middle of the day, when probably the activity of the insect is much, much lower. But if you involve night or evening or morning, no way, the mate. So the system should be on during the evening, during the morning, and the night. Okay. So basically, this was also another important information for us. And this is, however, the present solution that is say derived from all this information that we collected during the years. This is the vineyard in San Michele. This is San Michele all'Adige in Fondazione Mach, where I work. This is the first system. And here, electricity was given by the cables, so it was easy. This is another vineyard where we have solar panels. And solar panels mostly work in the day. With the battery, you can extend also for hours in the night. But here is the evolution of different types of prototypes. In the beginning, we started by applying this to the wire, but it was not a good solution. The efficacy was much, much lower. Then we moved to the poles with different types of prototypes. And this is the last one that is exposed outside at the table of BioGuard. And there is a solar panel for each of them. So they are standalone instruments. And they don't need cables or big solar panels in the vineyard that are a disturbance also for the farmers. They stay in the line. So this is what we aim to, that is the use of behavioral manipulation, putting together semiochemical, semiophysicals, and targeting different types of insects. Because the problem is that if there is one insect that can't be managed by behavioral manipulation, you need to use a pesticide. And if you use a pesticide, you say, oh, why should I use behavioral manipulation? I used a large spectrum pesticide. It's cheaper. So it's very important to find behaviors and behavioral manipulation techniques that are applicable to the main pests in vineyards, but also in other systems. And time is it? Yeah, I'm almost in the end. A couple of slides. This is another. These are the solution of semiophysicals, colors, but here these are the putting together semiochemicals and semiophysicals. Here you have a bait and the color. Here you have pheromone and vibrations together, because as you know, stink bugs use pheromones for large range communication. And I show you a video. This is Brown Marmorated Stinkbug, and it's a bimodal communication, pheromones and vibrations. In this video, you see that when we activate the vibrations, you see that this is a very infested tree. There is a behavior that is a call and stop, walk and stop. You see the animals, these are males, all males. They walk and stop, walk and stop. Because when they stop, they get the signal, and then they take this directional decision. Yeah, that plant was badly infested, but the point is to put them inside the trap. So, the signal is this. It's almost 80 hertz of frequency, and it's the female calling song, and males are very triggered by this. Okay, so this is the vibrational trap, which is also outside, and it works with a multimodal system. You can see that you have a solar panel here on the cap. Then this is the pherodone, I would say, dispenser at this point. That is the vibrational emitter of the female signal. Then you have a pheromone. So you have pherodone plus pheromone together. These are stripes that are inviting the insect. So the pheromone is for the long range attraction, and the pherodone is for the short range attraction. This system is giving us very good results, and it's probably the first example of bimodal practical application in farming. So I want to close with this sentence that was said by Piero Angela, which was the most important scientific journalist in Italy, and invented the science documentary in Italy. And he died a few days ago, and they say that knowledge of a person is valuable if it can allow him or her to understand the world where he or she lives, and possibly to change it to his advantage. That makes sense, because if we don't put in practice what we do, our knowledge is less valuable. It's not unvaluable, because somebody always can take your knowledge and transfer it. But if we can do by ourselves, it's something even better. And yeah, my last is for thanking many, many people that in the years and projects that in the years worked with us, San Michele and Fondazione Mach. And yeah, I want to especially thank Meta for hosting and the NIB crowd, of course, hosting this third Biotremology meeting. And thank you for your attention.